Welcome back. This is Lecture 29, Yeast and Mold Testing. The quantitative logic here is exactly the same as Lecture 28, dilute, plate, incubate, count, calculate. That underlying framework does not change. What changes is everything around it. Fungi grow slower, they spread differently, and they need a completely different medium to count accurately. This lecture covers exactly what changes and why, and just as importantly, why cutting corners here can let mycotoxin-producing molds slip past completely undetected. Here is what we will cover today. Number 1, you will understand why fungi need a dedicated method, rather than simply being folded into a standard total plate count. Number 2, you will know why DRBC agar replaced older acidified media as the standard, and what each of its ingredients actually does. Number 3, you will understand why spread plating is preferred over pour plating specifically for yeast and mold work. Number 4, you will be able to distinguish a yeast colony from a mold colony by appearance, and apply the correct countable range. Number 5, you will be able to connect a positive mold count back to the mycotoxin risk we introduced in Lecture 25, and know when further testing is actually warranted. Let's start with why fungi can't just ride along on a standard total plate count. Recall Lecture 25, yeasts and molds are eukaryotic, structurally different from bacteria, and it turns out they behave differently on a plate in ways that genuinely break the assumptions a TPC depends on. First, they grow slower. Fungal incubation runs a full 5 days, compared to 48 hours for bacteria. If you read a plate at 48 hours expecting fungal growth, you would simply miss most of it. It just is not there yet. Second, molds spread rather than simply multiplying in place. Fast-growing, spreader, molds, Rhizopus is a classic example, can overrun an entire plate, physically burying slower-growing organisms before you ever get the chance to count them. Third, fungi tolerate conditions that bacteria genuinely cannot. Low pH and low water activity let fungi thrive in jams, dried goods, and acidic products, exactly the kind of product where a standard TPC would show almost nothing at all, even if real contamination is present. This is exactly why yeast and mold testing gets its own medium, its own incubation conditions, and its own countable range. We are going to cover all 3 of those in this lecture. Let's talk about the medium, because this is where the method genuinely diverges from what we covered in Lecture 28. The current standard medium is dichlorin rose Bengal chloramphenicol agar, DRBC. It replaced older acidified media because acid actually stresses and injures fungal cells, which reduces recovery rather than helping it. Every single ingredient in that name is doing a specific job. Chloramphenicol is an antibiotic that suppresses bacterial growth, so bacteria do not compete with or overrun the fungi you are actually trying to count. Rose Bengal is a dye that yeast colonies absorb, turning them visibly pink, and it also mildly restrains colony size, which helps with counting later. And dichlorin specifically inhibits those fast, spreader, molds we just talked about, organisms like Rhizopus and Mucor, keeping their colonies compact instead of letting them overrun the whole plate. One exception worth knowing: for foods with water activity below 0.95, dried goods, spices, low moisture products, DG18 agar is used instead. That's dichlorin with 18% glycerol. Its lower water activity better matches the actual product being tested, and it further discourages both bacterial growth and fast mold overgrowth. Now let's talk about plating. Recall Lecture 28, pour plate embeds colonies in agar, spread plate keeps them on the surface. For yeast and mold work specifically, spread plate is the preferred technique, and there's a real reason tied directly to how fungi grow. Here's why not pour plate. Embedding fungal propagules in molten agar, which sits around 45 degrees Celsius, can thermally stress or even kill fragile fungal cells before they ever get the chance to grow. That undercounts the true population. Here's why spread plate works instead. Diluted sample is pipette bound to solid, room temperature DRBC agar, and spread evenly across the surface. There is no heat exposure at all, colonies stay visible on the surface where you can actually see them. And the dichlorin in the medium keeps fast growers from taking over before you get to count. One more thing worth knowing: for certain non-liquid foods, nuts, spices, whole berries, There is an alternate technique where surface disinfected food pieces are placed directly onto solidified agar, rather than diluting and pipetting at all. It's a good reminder that method selection always starts with the actual product sitting in front of you, not just a default habit. Let's put yeast and mold incubation side by side with the TPC conditions from Lecture 28, because the contrast really drives the point home. Medium, DRBC agar, or DG18 for low water activity foods, versus plate count agar, which is non-selective, for TPC. Temperature, 25 degrees Celsius, plus or minus 1, versus 35 degrees Celsius, plus or minus 1, for TPC. Duration, 5 days versus 48 hours, plus or minus 2, for TPC. Light exposure, incubated in the dark, because rose Bengal degrades under light, versus no special light requirement for TPC. Plate orientation, right side up, not inverted, and stack no higher than 3, versus inverted, lid down, for TPC. And countable range, 10 to 150 colonies per plate, versus 25 to 250 for TPC. Every single one of these differences traces back to something real about how fungi behave differently from bacteria. None of it is arbitrary. Now let's talk about actually reading the plate, and telling yeast and mold apart at a glance. Yeast colonies. Smooth and raised, moist looking. Pink to reddish in color, from that rose Bengal uptake we discussed. And they have discrete, well-defined edges, counted much like you would count a bacterial colony. Mold colonies. Flat or fuzzy, spreading in texture. Natural pigmentation coming from their spores, which can be green, black, or white depending on the species. And here's an important detail: diameter matters. You count mold colonies according to method guidance even if some merging has already begun, unlike with bacteria where merged colonies typically get excluded. The countable range here is 10 to 150 colonies per plate, narrower than the 25 to 250 range for TPC, because fungal colonies simply take up more space and merge sooner. Plates outside this range get reported as estimated counts, exactly the same convention as with TPC. Now let's look at what this actually looks like in practice. Take a look at the 3 photos on this slide. First, yeast colonies on DRBC smooth, raised, pink colonies. Second, mold colonies on DRBC, fuzzy, spreading colonies with visible spore pigmentation. And third, a mixed plate showing both colony types together, which is genuinely the most useful reference, since it lets you compare the two side by side on the exact same medium. Once you have seen yeast and mold colonies together on the same plate, telling them apart on a real bench sample becomes almost automatic. Let's talk about why a mold result can't just be filed away once you have your number. Recall Lecture 25. Some molds, certain Aspergillus species in particular, produce mycotoxins that persist even after the mold itself is gone. A mold count is never purely a spoilage number. It can be a safety number too. Let's walk through typical next steps based on the result. If the count is within specification, that's routine trending. No further action needed beyond standard record keeping. If the count exceeds specification, that triggers an investigation into the source, plus a review of storage conditions and water activity control. If you see visible Aspergillus-type colonies, you should consider mycotoxin screening. Aflatoxin testing, for example, even if the overall count is within limits. And if you see repeated exceedances in one specific product line, that calls for a full root cause investigation, which often traces back to humidity, packaging integrity, or ingredient sourcing. Here's the core point to take away: a mold count is a screening tool. Species level identification and mycotoxin testing are separate, targeted follow-ups. The count itself does not provide either of those on its own. Before we close, let's walk through where yeast and mold counts commonly go wrong. The first mistake is reading the plate too early. Fungal growth is genuinely slower than bacterial growth. Reading before day 5 will underreport a real contamination that simply hasn't shown up yet. The second mistake is leaving plates exposed to light. Rose Bengal degrades under light exposure, forming compounds that can actually inhibit growth and skew your count. Keep these plates in the dark. The third mistake is applying the TPC countable range out of habit. 10 to 150 is the correct range here, not 25 to 250. Using the bacterial range will misclassify plates that are actually perfectly reliable. The fourth mistake is assuming a low count means no mycotoxin risk. A small handful of toxigenic mold colonies can matter more than a high count of completely harmless ones. Count and species identity are two separate questions entirely. And the fifth mistake is using pour plate out of habit. Heat from molten agar can injure fragile fungal cells. Spread plate is the preferred technique for this method specifically, for the exact reasons we discussed earlier. Let's bring this lecture to a close with the key takeaways. Number 1. Fungi grow slower and behave differently on a plate than bacteria, which is exactly why they need their own dedicated method. Number 2. DRBC agar uses chloramphenicol to suppress bacteria, rose Bengal to mark yeast pink, and dichlorin to stop spreader molds. Number 3. Spread plate is preferred over pour plate here, because molten agar heat can injure fragile fungal cells. Number 4. Incubation is 25 degrees Celsius for 5 days, in the dark, plates right side up, all different from the TPC conditions in Lecture 28. Number 5. Countable range is 10 to 150 colonies, narrower than TPC's 25 to 250, because fungal colonies take up more space. Number 6. A mold count is a screening tool, not a mycotoxin clearance. Toxigenic species need separate, targeted testing. That is Lecture 29 complete, and with it, the core quantitative toolkit for this level is done. You now know how to run and read a yeast and mold count from start to finish, why its conditions differ so deliberately from TPC, and why a mold result can trigger mycotoxin follow-up even when the count itself is low. Up next is Lecture 30, Coliform and E. coli testing. That lecture shifts us toward indicator organisms, a slightly different logic than either of the last two lectures, where the organism you count is standing in for a broader hygiene or contamination signal rather than being the direct concern itself. Take a look at the roadmap on this slide. Lectures 24 through 28 are done. We are wrapping up Lecture 29 right now, and 6 more lectures remain in this level. If this lecture helped you, please give it a like. Subscribe for new lectures every week. And drop your questions in the comments, they get read and answered. See you in Lecture 30.